Yellow. So this morning I have a bunch of stuff to do. It's like going to be a long day. So remember, I am interested in evaluating the influence of light conditions and nutrition. So the different media type on growth and sporulation of a fungus that I'm going to be working with. And so today is the assessment part of it. I have to harvest those spores and then I'll have to count them. So I think I have 27 treatments. So I have to count them and then compare them. So that's what I'm going to be doing. The BSc is currently on its UV first secondary sterilization, and then I'm going to go to the different incubator that has the light conditions to collect the plates, and then I'll get into it. So I'll be using the spectrophotometer today to do concentration reading. Then these are the fungal samples after 14 days, 2 weeks. So I have 3 different pathogens, 3 different media type or nutrition, and 3 different light conditions. So the first row are continuous light for the 14 days. Second row, continuous darkness for the 14 days. And then the last row is a 12-hour light dark cycle. And this already has sclerotia. Those black structures, they are survival structures. So this pathogen can stay in the soil or in the parts that it infects for a long time by using those hard structures. So I'm going to turn on the BSC, like take it out of the UV cycle, and then start working. It is 8 o'clock, and let's see if I'll finish before 4 or 5. Inside the BSC, I have the plates that I sterilized before putting them in, and I'm always collecting observation data. It's important, so I do that using photos. So I'm setting up my Teams chat that I load my pictures in. I take a photo of the base of the plate, then I open it and take a photo of the actual colony, and I hold them up, ensuring that I'm getting the right angles, and I'll just keep repeat doing that until I photograph all of the images. Some of the plates you can see that they have different colors, and it's just that the different pathogen produce different colors for different reasons. Some of the darkness or the pigmentation helps them to infect. After I've collected images, I move in cycles. So if I'm doing light, I do light first. If I'm doing A, I do A first. So here I am carrying out the spore harvesting. So I sterilize that little device, that little glass rod with the black thing at the end. It's called a policeman. I pour water and I use the rod to dislodge the spores. So you can see the white mycelium is coming off. Those hard structures are sclerotia, and the pathogen use them to survive during unfavorable conditions. Once I'm done, I pour that off into a tube, add some more water, top it up. Then I filter that spore suspension using sterile cheesecloth and the filtrate, so whatever comes out of it, that now becomes the spore suspension that I'll use to count the number of spores and to see which light conditions and which nutrient source increase sporulation. I always listen to music when I work. I'm always dancing. It's like the soca I'm listening to. I like listening to soca because just the beat, the rhythm, it just gets me to power through, especially on those long days. I can't remember what song it was, but I always take breaks to stretch because I'm just sitting in that position for such a long time and I'll end up being sore. So enjoy. In the next few clips, I will be going through the spore harvest process again because I have 3 sets. I take images. That is my observation. I'll use it in my report. Once that's done, I'll use a sterile rod, hockey stick, not like a real hockey stick, just an L-shaped glass rod, or that policeman to scrape the water that I add on the plate. The water will collect the spores that I'm dislodging. I will strain or filter that into a new tube using sterile cheesecloth. Once that's done, I top it up, sterilize my policeman again, label my tubes, and then put it to the side, because now the tube that has the strained liquid in it will be the one that I'll use for assessment. So that's what I'm doing. So I've finished harvest the spores. That's them in water. It just looks like cloudy water or dirty water. So up next, I'm going to measure their concentration using the spectrophotometer, and then I'm going to look at the spores here under the microscope so that I can do a spore count. Let's see, it's 1:30. I am tired. I'm going to aliquot 1 ml or 1,000 microliters from the tube with the red top into each of the tubes with the orange top. In the appropriate tube, I have them lined up so they match these. And once that's done, I'm going to put them into cuvettes. Oops, it's not focusing. Yeah, into cuvettes. It's already set. Blanket and take the reading. And this is the hemocytometer, and I'll put a drop here in one chamber, a drop there, put the slide on, and then look at the spores under the microscope. I'm going to take a picture and then count them at home because I'm tired. So I'm finished transferring them into these 2 ml tubes, and I'm going to put everything into this. This is a cuvette, and then take the reading. So I'm going to use spectroscopy via the spectrophotometer, which is the machine, to note the concentration of all the samples based on nutrition and light conditions. So, the spore suspension. So I have to blanket with water. So just to get a 0 reading, once that's done, then I will load all the samples one at a time and record what the concentration of the spore suspension is, and then use that further in my report and my presentation. So now I'm going to add acid fusion. It's an acidic dye. Same process, it stains the cell wall of the fungus pinkish peachish. So I'm just going to put them, I think, 100 microliters into each of these vials, and then let them incubate before I look at the spores. It is after, what's that, 3? God, I'm tired. But I am going to take 10 microliters from each of these. I have 27, and I'm going to put it on the hemocytometer. There are 2 chambers, and take a photo of both chambers for each of them. And then tomorrow or Friday next week, I'll go through and count so I can compare across the 27 which has the most and stuff like that. Yeah, it's not focused, but that's a counting squares. Magnified, and then you count the cells. You select one and then count the cells in a square.
The spores are the small circles.
It's 5:57. I came in at 7:30 a.m. Finally done. Well, for the in-lab portion. And then tomorrow or whenever, well, tomorrow I'm going to call it a short day, but in the coming days I'll do the count of the spores to see the influence of the light condition and the nutrition on the number of spores produced by the 3 fungal isolates. But I am tired, in need of a massage. I should book one. But ciao!